The connectome, mapping every neural connection in the human brain, how scientists attempt to chart the brain's complete wiring diagram, why mapping the brain became a scientific goal. For decades, neuroscientists have tried to understand not just individual brain regions, but how everything connects. The brain is not a collection of isolated parts. It is a network where billions of neurons communicate through trillions of connections. Understanding this network is essential for explaining how thoughts, memories, and behaviors emerge. This goal led to the concept of the connectome, a complete map of all neural connections in the brain. It represents an attempt to move from studying parts to understanding the full system. What the connectome actually represents. The connectome is often described as the brain's wiring diagram. Just as a map of roads shows how cities are connected, the connectome shows how neurons are linked through synapses. It includes both structural connections, which describe physical links between neurons, and functional connections, which describe patterns of activity between regions. Together, these connections form the pathways through which information travels. Mapping them provides insight into how the brain processes and integrates information. The scale of the challenge. The human brain contains approximately 86 billion neurons, each forming thousands of connections. This creates an incredibly dense network. Capturing every connection at this level of detail is a major technical challenge. Even mapping a small portion of brain tissue requires enormous computational power and data storage. Scientists often begin with smaller systems, such as the nervous system of simple organisms, before attempting larger and more complex brains. How scientists collect connectome data. To build a connectome, researchers use advanced imaging techniques. Electron microscopy allows scientists to capture extremely detailed images of brain tissue, showing individual neurons and their connections. These images are then processed using computational tools to reconstruct networks in 3 dimensions. Other methods, such as diffusion imaging, help map larger-scale connections between brain regions in living humans. Each method provides a different level of detail. Reconstructing neural networks step by step. Creating a connectome is not just about taking images. It involves reconstructing connections from massive datasets. Scientists must trace the paths of neurons across thousands of images and identify where synapses occur. This process often requires both automated algorithms and human verification. The result is a digital model that represents how neurons are connected. Even small errors can affect interpretation, so accuracy is critical. What connectome mapping reveals? Mapping neural connections helps researchers understand how information flows through the brain. It can reveal patterns such as highly connected hubs or pathways that link distant regions. These patterns are important for understanding functions like memory, perception, and decision-making. Differences in connectivity may also explain variations in behavior or cognitive ability. The connectome provides a framework for linking structure to function. Applications in neurological disorders. Changes in neural connectivity are associated with many neurological and psychiatric conditions. Disorders such as Alzheimer's disease, autism, and schizophrenia may involve disruptions in network structure or communication. By comparing connectomes from healthy and affected individuals, scientists can identify differences that contribute to these conditions. This could lead to earlier detection or more targeted treatments in the future. Limitations of current technology. Despite significant progress, mapping a complete human connectome remains extremely difficult. Current methods either provide high detail over small regions or lower detail over larger areas. Combining both scales into a single comprehensive map is still a challenge. Data processing and storage requirements are also enormous. These limitations mean that current connectome models are still incomplete representations of the full brain. The future of connectome research. Advances in imaging, artificial intelligence, and data analysis are expected to improve connectome mapping. Automated systems may speed up the reconstruction process and reduce errors. Larger datasets will allow scientists to compare connectivity patterns across individuals and populations. Over time, this research may lead to a more complete understanding of how the brain's network structure supports cognition and behavior. Final thoughts. The connectome represents one of the most ambitious goals in neuroscience. By mapping every neural connection, scientists aim to understand how complex mental processes emerge from biological networks. Although the task is technically challenging, progress continues to reveal new insights into brain organization. The connectome shifts the focus from isolated regions to interconnected systems, offering a deeper understanding of how the brain functions as a whole. Thanks for watching.